Now we are live on YouTube, and I forgot as well to say we are broadcasting this live. Sophia, agree. If you are here and you don't want to show on YouTube, so don't turn on your camera and don't speak. But Sophia, one more thing, like what about the questions? How do you prefer us to handle the questions? Do you now we are live on YouTube, and I forgot as well. Do you prefer questions on demand or leave for the end? What do you prefer?
Well, if there is something that, of course, you are not understanding at the moment, yeah, you can ask right away. If not, of course, I left time for us to discuss in the end anyway. So I'm comfortable either way.
Okay.
As long as you manage the questions, because I will be like without being with access to the chat or something.
Yeah. So and we try to keep things as we used to. Like you are free to make questions in a civilized way. I won't be here like giving the token. So if you want to make a question, you can put on the chat if you're shy or if you want to just speak up. And for those who are on YouTube, you can write on the chat there, and I'll bring the questions here. Okay, so it's already too much of introduction. Sophia, thank you again. The floor is yours. Thank you.
Thank you very much. Let me share my screen. Okay. Okay, so you are able to see my screen.
Yeah.
Yes, perfect. So thank you very much, Diogo, for the invitation. Thank you all for being here with me today. As Diogo said, I'm an assistant professor now in the Department of Informatics in the University of Lisbon. I also took my PhD there in Technical. And today I'm kind of reviving a little bit the work that led me to actually enter the network science community. I started really as a computer scientist working on efficient data structures and so on. And then at some point, I surrendered myself to the wonders of network science. And so I will be talking about link significance in phylogenetic analysis and diversity in networked populations. So basically, my interest during my PhD was actually then to study this kind of emergence in complex systems and even how entities or how these dynamical systems self-organize over time. Today, I will be then focusing on structure and dynamics on networks, more connected than to bacterial populations, so more in an epidemiological side. And so I divided this presentation in two parts. One of them, the first one presenting an edge centrality, so an edge-based metric to measure link significance. This, as I will motivate this, arose from a biological problem. We published a couple of papers back then. And then I will jump to actually a problem that is even to have these links to analyze. Sometimes we need data that is constrained due to sample sizing and so on. So I will present theoretical work that we did based on some parameters that we discussed with our biology partners, in which we study like how network structure plus the evolutionary dynamics can then impact the diversity that we find in certain populations. And of course, we introduced this kind of heterogeneous network contacts that usually in biology, most studies in the beginning at least assume like a well-mixed population. And so we introduce here that heterogeneity between different host populations. But let's dive into this first part of link significance. So the use of phylogenetic representations actually started like a long ago, more or less in the middle of the 19th century. And of course, one of the most popular uses is the Charles Darwin sole illustration in the origin of species. And of course, these trees, they offer a simple representation, but it still makes a good approximation, a good choice to convey the diversification and the relationships between species. So nowadays, actually, with the large availability of sequence-based microbial typing methods, as for example, the multi-locus sequence typing, minimum spanning trees have become like the standard for representation of relations between strains. And of course, each algorithm or method that is used to infer or draw a tree makes a series of assumptions. And so they make these assumptions that the conditions that arise from building these trees are important then to have some kind of biological ground. But of course, since we can have like multiple trees, it's really difficult to evaluate what is like the statistical significance of the trees that are presented. So what we are proposing here is that we provide now this new statistical metric to evaluate the importance of the links that we observe in these trees. And so just, of course, I think that most of you are really comfortable already with the concept of minimum spanning tree, but what is a minimum spanning tree? So given a weighted graph, is the minimum spanning tree is then the subset of the edges that connect the entire network, but which has the least weights connecting each pair of vertices together. So we have like the minimum possible total edge weight connecting the entire network. And one of the things that we will need, of course, to calculate this measure to evaluate minimum spanning trees is then the problem of counting minimum spanning trees. And for unweighted graphs, this problem is well solved with the Kirchhoff's matrix theorem. For weighted graphs, it's not so simple, but we will see how we solve this problem. And so to calculate even the number of minimum spanning trees where a given edge appears, so for us to say what is the probability of an edge to be in a minimum spanning tree, as far as we know, at least at the time, was the first approach. And so we define this new measure as the spanning edge betweenness, in which we will then calculate the number of different minimum spanning trees for a graph where a given edge occurs. And then it's just a simple division with the total number of different minimum spanning trees for this given graph. And as I said, for undirected and unweighted graphs, of course, I'm assuming here that we have undirected graphs. We can use then the Kirchhoff's theorem, in which we basically remove one line and one column from the Laplacian matrix. We calculate the determinant, and so we obtain the total number of minimum spanning trees for a graph. And so to calculate then the number of minimum spanning trees that a given edge is present, what we do is to remove both rows and columns that correspond to the indices of the vertices of the endpoints of the edge that we are calculating. So for example, here, if I want to calculate the number of minimum spanning trees that this edge here connecting 0 and 1 is present, I go to the Laplacian matrix, I remove both rows and columns of the indexes 0 and 1. I calculate the determinant of the matrix, and I get the number of minimum spanning trees that this edge is present. And so if I want to obtain the spanning edge betweenness, this is the fraction of minimum spanning trees that the edge is present. I just need to divide these two values. But what happens for connected, undirected, but weighted graphs? So our methodology here was to, we will do similar calculations, but first we need to sort the edges in increasing order. And so after sorting the edges in increasing order, Then for each weight, we will create all possible connected components containing the edges with that weight. So it's like we will process weights by level. And so for each given weight, we will create then the connected components containing the edges. And then for each connected component, we apply again the Kirchhoff's matrix theorem. So it's like if we divide this in different levels, in each level, it's like we have an undirected, unweighted network. And so the way that we update the total number of minimum spanning trees in the network is then by multiplying the number of minimum spanning trees of the network at the time with the number of minimum spanning trees of the components. And then after this step, we can compute for each edge the spanning edge betweenness value using then the formula that we just saw. And so in the end, after going to the next level of weights, what we need to do is to contract each connected component in one node. And if we have more than one component left, of course, we go to back to step 2, so for the next weight. This way, then we are able to compute also the spanning edge betweenness for weighted networks. Given that we are then in a computer science department seminar, I leave here some details about implementation. Of course, implementation is publicly available in the papers. We have the links for the Bitbucket repository. And so to compute the determinants, we use then the LU decomposition to accelerate on top of LAPACK. And then instead, of course, of multiplying all the determinants, because at some point we don't have any space for them, we use then the sum of the log of the determinant. And to compute the connected components, we use then a disjoint set data structure. And in terms of complexity, of course, to compute the determinant of a square matrix, this can be done in N32. And so for sparse graphs with the number of edges being close to the number of nodes, then this method runs in ON 2.5. And so, of course, this increases because we have to compute the determinant for each edge. So in practice, it runs faster as connected components are usually smaller than the original graph. But the first result that I want to present has to do more for us to be aware that actually, even for small data sets, so I put here some of the bacterial data sets that we were studying at the beginning, that the number of minimum spanning trees actually, of course, rise much, much fast. And at some point, we really need to then have like this measure. So just reinforce that we need this measure to evaluate the significance of the links that we observe. This column here regarding edges without ties, as we will see, are the ones that have spanning edge betweenness equal to 1. This means that there is like only one connection between two of the nodes. But of course, as the network increases the size and even the number of edges, the edges without ties don't increase so much, but the number of minimum spanning trees increases a lot. So what we do is, of course, just graphically illustrating what happens is that if I have then, for example, the second largest clonal complex, that is how in biology they call the connected components. We have then the graph, and then, for example, if in the end the algorithm presents the spanning tree for that same graph, what we can obtain is then this ordered list of the frequency, so the percentage of minimum spanning trees that a given edge appears. And of course, as we see, for some of these edges, they appear 100% of the times, which means that that specific connection is the only evolutionary explanation for the strains to arise. So for example, as this one here, 79 to 80, here this, actually, no, I'm wrong, because there is a yellow guy here that even in my screen doesn't I see so well, so apologies for that. In the end, we provide not only this list, but also in the graphical format, we also provide, given the initial clonal complex, then also the percentages next to the edges. And so with these visualizations, we are able to at least assess if the phylogenetic tree that we see for a given species has a lot of confidence in the edges that it presents, and also because this could be used to also compare, for example, with bootstrapping algorithms and see that we may have in this way at least a statistical metric to analyze and provide a confidence for the phylogenetic tree that we observe. Of course, the spanning edge betweenness then in this way can be also faced as a measure to quantify this kind of link significance, but what else besides phylogeny? So I took the liberty of just providing this out of context slide in the sense that a little bit later we used this same measure to test, for example, then the resistance in network connectivity to the removal of edges using the spanning edge betweenness. So it's just an evaluation of if I calculate the spanning edge betweenness, and for example, we compare then with edge betweenness and random removal. If I calculate then the spanning edge betweenness, the edge betweenness, I sort in decreasing order, and now I start removing these edges of the network, what happens to the number of components? So what happens to the connectivity of the network? And we used two real data sets that we took, I believe, from Leskovec website, and also some artificial networks that we generated, some with specific community structure, and also with Barabaji and Albert methodology. And so what we observed is that actually for all networks, if we remove the edges with the spanning edge betweenness, compare then with edge betweenness, the blue line, or random removal, that we kind of break the networks into more components faster than with the other approaches with edge betweenness and random. And so this also gives us a hint that we still are investigating in how actually the spanning edge betweenness can also be useful. And I think that I say that in the next slide, to study this kind of network properties in, for example, epidemic spreading phenomena and so on. But of course, then before going from this micro level to the macro level of the populations of bacteria, I want to then just to reinforce that we provided then a new edge centrality, the spanning edge betweenness, again, that is defined as the fraction of minimum spanning trees containing a given edge. And of course, here the intention and the mindset was all about confidence. So this measure in the phylogenetical context allow us to decide and even have more confidence or less confidence in the proposed tree as a phylogenetic hypothesis. Of course, in the more biological part and analyzing better the trees and these percentages, we are also able to study a little bit more what is the impact of recombination and mutation events. So if the origin of a given strain is clear or not. And of course, then on this other part, expanding the application of spanning edge betweenness to other types of networks and applications. We think that, of course, this metric helps us to identify edges that are critical to keep the network connected. And of course, this may help us to analyze then what is the expression of the redundancy and alternative network configurations for a given network. And so I'm now going to the second part. I don't know if you have any questions, but if not, we can have them in the end. Okay, so if no one is asking anything, I will continue. So the second part is then, of course, even for these phylogenetic studies and even for these epidemic outbreaks, we wanted to investigate what is the impact of network structure in biological diversity using then large-scale simulations of bacterial populations over host contact networks. And what is the motivation behind this work is, of course, that we believe that understanding bacterial species population structure and how it can be shaped by genetic force of mutation and recombination is vital, of course, to interpret the response of bacterial population to selection pressures, and also to be useful to interpret the response, for example, for antibiotic treatment or even vaccination. So these large-scale studies are then important to understand this process, but also to validate, for example, models and methods as phylogenetic inference algorithms, as we just saw, and to understand then how the sampling that is commonly done can affect or bias the perception of the true population structure. So the observed population structure, of course, of several important human pathogens can be explained using a simple evolutionary model based on neutral mutational drift and modulated by recombination. I will provide a couple of more details next. And as I said, of course, validation of methods of phylogenetic inference and so on with real populations may not be possible if sometimes we only have like small samples of the real pathogen population that are available and accessible. And so how can we obtain these larger samples of bacterial populations? Are all populations well mixed? What is the impact of these host contact network topologies and associated transmission ratios on bacterial population evolution and genetic diversity? And again, we have even one more question, that is, what happens in the absence of selection phenomena? So what I will do is that besides presenting the evolutionary model and even how these interactions between different host populations happen, we will then analyze the impact of the host network topology on the bacterial population genetic diversity for different rates of mutation and recombination. And so we are interested to see if strain persistence and local evolutionary events that will happen in each population can then exchange due to the host contact network, and how the variations of the diversity may or may not be attributed to selection phenomena in the absence of having a selection pressure in the model. And so, of course, we need simulations for this. We are using large graphs to represent interactions between bacterial populations. So in these networks, each node is a population of bacterial, and each link represents then a connection between populations in which there will be the genetic material transfer between the different populations. Of course, if we want to do these large-scale simulations, they demand heavy computational processes. So I introduce here another detail of implementation, that is, we use the MapReduce programming model with Apache Spark and GraphX to be able to do these simulations in a timely manner. But let me start maybe with the biological model. So we use the infinite allele model, so that is also known as the Wright Fisher model, in which we assume that genetic events do not contribute to the fitness of an individual. So all individuals in the population have an equal chance of reproducing and being subject to genetic events. And under this infinite allele model, it always generates a new allele, leading then to a new profile. And so what we see here is that if I have this population with sequences of size 3, so each one of these is an allele, each time I have a mutation event, I'm generating then a new profile that I didn't see before. But when I have a recombination event, so I'm transferring one piece of my sequence to another one, then it could be that we could have like strands reappearing again after a while. And so mutation and recombination in this model, they occur independently, with each event being, we can, well, depend on the rates that we use, but they are rare, and mutation usually takes precedence over recombination. And so a new generation is obtained at each step, leading to non-overlapping generations. And so what we do is, besides then to having the mutation and recombination events, we sample then elements from the previous generation to the next generation. How does it work then when we have an interaction model? So when we have a network with different bacterial populations, so each node is a population. Each population will have a given number of individuals. Each individual will then have a profile that is composed by alleles, as we just saw in the previous slide. And so we will have then mutation and recombination rates associated to each population in each node. Given a contact network, what we need for the network simulation process is, of course, the edge list with the structure of the network, and also with the timely frequency with which the populations will exchange material. And of course, the transmission ratios. So how much of each population will then move to another one. And what happens is that given then these transmission ratios, so what happens is that given the pool of individuals of a given population, when we receive the other genetic material of the other populations, in the end, we sample again to then have a fixed size population. And so at each time we have this exchange of material, this is the process that is happening. Regarding the computational model, so a little bit of detail, but we use then MapReduce for the evolutionary process in the nodes independently. So, and this is a two-phase programming model in which we have a key value, and for the value, we have some kind of transformation operation that is done independently into our data. And then we have a reduce phase in which then the results are aggregated for a given key, for example. And so Apache Spark is actually a very nice framework with a very nice interface, and I believe that is simple to use. And this framework extends the MapReduce model that enabled then the creation of iterative programs. So it's like you are developing your own program, but we are still able to easily parallelize the framework. And so, of course, with the resilient distributed datasets data structure, it's more resistant also to failures during the computational process. And we use GraphX to then shape the contact network, and this API allow us then to address these operations in a graph parallel manner using the same resilient distributed data structures as Apache Spark. So in the end, the simulation framework is basically like this. So we have then the evolutionary model on each node using just simple Spark commands, but then we also have the structure of the host contact network and the exchange process modeled with GraphX. And in the end, actually, this is just one single iterative composable API. And so for this experimental setup, we used at the time the Google Cloud Platform to run our simulations. So I have here some technical details that I won't go much further, but for the results that I'll be presenting, we run at least 2,500 generations on our simulations with cycles of 25 evolutions, so 25 processes of mutation followed by a recombination in a single population, followed then by one phase of interaction between the populations of the network. And then we compute the Simpson's index of diversity for each node to analyze then the diversity of the populations. So we set the population size as 1,000 per node, and each simulation would contain 1 million individuals, since we have also, our networks are also of size 1,000 nodes. And then we made combinations between mutation and recombination values in this interval. And so for the computational performance, actually, we were very happy because, of course, when designing parallel algorithms, it is important to estimate the relation between achievable setups and the number of workers. And what we observe here is actually that the speedup becomes more evident as the network grows in size and density. And for the click topology, actually, this led to the highest computational cost when performing the exchanges, because of course we have many more connections than in scale-free networks, or for example, this regular that I forgot to mention. So we used fully connected networks, we used the regular networks, and then we used 2 scale-free networks, one Apache and Albert, and one duplication model, this with degree 4. Our intention was then to model not only well-mixed populations, but also then heterogeneous populations. And of course, the speedups and even the decrease in processing time was much more visible when we had the fully connected network. And that is even more observed in when we observe, for example, how the scale up is done with these curves provided by the Amdahl's law, where the percentage corresponds to the fraction of the computation that is actually infinitely parallelizable. And of course, with the clicks, so with the fully connected networks, we achieve a more percentage than actually with the regular or the lower degree networks. But let's go to the results regarding the genetic diversity in the populations. So the Simpson's index diversity is a probability then, or can be said as the probability of 2 individuals randomly selected from a sample belonging to the same species. And this is for a small network that we tested before doing these large scale simulations. And we were curious from the beginning that for some nodes, actually, the diversity would decrease significantly, and without, again, having any kind of selection process. So we have mutation, recombination, and then just a sampling process. And for our results, actually, then for these large-scale simulations, again, we observed that, for example, on this left side, I have like the average Simpson's index diversity through time for the different network topologies. Of course, for big high rates of mutation and recombination, at some point the diversity increases and stay there. But for example, for the second part of the plot, I have the quantiles and the median for the Simpson's index diversity. And we can see that actually, so for the fully connected, it's more or less expected, since we have like these highly connected interactions, that at some point the diversity still converges somehow. But for the heterogeneous networks, or at least for those that are not fully connected and have a more heterogeneous structure, we can see that even for relatively high levels of recombination and mutation as 0.001, we still have nodes oscillating in their diversity through time. And of course, this is something that must further be studied, because one discussion that we always had with our team, with also molecular biologist researchers, was that most of the times, maybe when analyzing these results or observing this kind of oscillation in diversity of species, this could be then erroneously be attributed to some kind of selection events. And this suggests, of course, that we need really to distinguish between genetic drift and selection to understand natural evolutionary processes. And with this, I conclude my presentation. And of course, if you have some questions, I'm happy to answer them.
Thank you, Sophia. So while people jump on and make their questions, I have a very, very basic one. Try to be like to help those who are not very familiar with networks and all this stuff. So go back to the basics. Like you talk in your first the first part about minimum spanning trees, and then you introduce this algorithm. So why should we care about minimum spanning tree? Like, what does it give to us? Do you have like examples of applications and your spanning edge between this is something that we can use to measure, for instance, the shortest path, or it's something that we find the resilience of the network, or something, or no, to find the importance, the centrality. Can you talk a little bit about these things?
Yes, I'm really happy that you asked that question, actually. So I think that at the time, I'm sure the motivation was that when analyzing other edge centrality measures, as edge betweenness, cut centrality, and so on, they actually provide information more about the flow of the network than actually about the structure of the network, or at least in my interpretation of those measures. So at some point, I think that the question was like finding a measure that actually could be topological, giving information about the topology per se of the network without necessarily being then shortest path or something that would involve the flow of the network. And so what hopefully I have now a master student that is picking on my work and starting to explore a little bit more of this, is precisely for us to also know. So I did that preliminary analysis, like that very small analysis on just testing, okay, if I want to test, for example, resilience of the networks, imagine a transportation network, a power grid network or something like that. And I want to identify, for example, weak connections or something, because I know that even in percolation problems, we usually use more a node-centric approach in which we are removing nodes or so, testing how the network still keeps connected or not. But actually, sometimes we cannot destroy the places. We can maybe interrupt the connections or something like that. So I thought that there was space still to investigate further other types of edge centralities. And on this case, pure topological measure. Well, I know topology has different meanings in different fields, but in this case, in terms of structure of the network. And so even, for example, for epidemic processes and so on, hopefully we will investigate at least how this could be also useful and helpful to maybe identify even locations on the network for interventions or something like that. But it's still a work in progress for sure.
So related to this, like Marcos just asked about if this approach could be used to investigate the dynamics spreads in the network. So
yes. Yes, I believe that even there was recently a paper that I think that cited us, and actually it's fun because, for example, one of them was about watershed networks. So a lot of things that actually you still need to somehow keep some kind of robustness in the connections of your system and so on. But here, in the social context, I'm also very curious about how this could then actually be useful to give more information about these opinion dynamics and spreading processes, of course.
I don't know if it's related or not, but something I was just thinking now when you're talking about like the evolution and this difference between topological or flow of things. Like maybe it's something that we should think about the problem. If the problem usually evolves to the best path in terms of more efficient, shortest, or whatever, or if they evolve through the most common, most easy, most resilience in sense of that we have many repetitions of the same, and it's not because it's the best, but it's just because it's the easiest to find or something like that.
Yeah, and also because, for example, depending on the structure of the networks, it's also like a direct measure of, for example, if I find a link with spanning edge between us one, I know for sure that if I remove that edge, I break the network. So it's a consequence for sure. What happens in the range between, well, if it's zero in a weighted network, of course, I'm not in principle breaking the network, but let's put the simplest scenario in unweighted networks. I will always have then values greater than 0 and up to 1. If it is 1 and I remove the edge, I break the network. The other range of values, of course, on this initial analysis, we saw that still we break the network faster, but we still want to investigate further what specific intervals means in terms of then robustness and resilience in this context, because we were not yet able, well, actually, I didn't have the time yet to investigate that, if specific intervals of the measure are actually mean some specific part of the process of these networks. And so, yeah, I think that it could be nice to even identify then bridges or even these important connections, as Federico was saying, for example, in social networks. So yeah, hopefully now, and I think that this is the greatest advantage of being a professor now, is that I will have more people to help me to investigate further these things. So yeah, I'm really excited about this.
Hugo.
Hi, Sophia. Thank you. Thank you a lot for the talk. Really interesting. Sophia, my question is more on like, let's say it's more like a philosophical, futuristic kind of question. But one thing that happens with the introduction of these new paradigms, in these new computational paradigms in different areas, that it's not only that they provide us faster or better solutions for existing problems, but also they allow us to find solutions for new problems that we didn't have solutions before. My question is, how do you think these large-scale simulations that you tested with Spark and MapReduce, what kind of new, let's say, findings or phenomena it can help us to uncover because of the larger scale of the simulations that they enable us to do?
Yeah, so, well, that is also a great question in the sense that, for example, I remember when I started developing the simulator, at the time, I was not yet doing any kind of parallelization and so on. And for one set of parameters, I would like wait hours, if not sometimes days, to get the results, plus the analysis and so on. Thanks to these frameworks, and now with the increasing availability of this cloud computing, I was able to like have this in seconds. So for a bunch of parameters, and of course, because I did, I think it was at least 1,000 times, so the average and so on. So for each set of parameters, if you have, so this is exponential, of course, the number of experiments that you need to do. And for example, now we are in the midst of COVID and so on. And if you want to do even this large scale and to have some kind of simulations, or I'm not talking about even forecast, but even to try to analyze or something and to reproduce in a high scale resolution, it's impossible to obtain results in the same day and to provide. And even, for example, this was never implemented as an interface for biologists, but of course, no one likes to wait a lot of time to run this stuff. It's the truth. So I think that fortunately, this will be something that may help for immediate response or even for some kind of emergency situation. These technologies, I'm not even talking about my simulations, but the fact that we have these that allow us to simulate on networks in this really, really fast way, I think it can then give more options for any kind of application that may use networks. And because I simplified a lot, because GraphX and Apache Spark, they allow you to have like attributed networks. They allow you to have like a lot of information that I didn't use because I managed to simplify a little bit more for the sake also of being fast and getting the results. But yeah, I really think they can change a lot in terms of timely response of problems.
Thank you, Sophia. Thank you.
Any other questions? I don't see any question here on YouTube.
Yes, I have a question. Do we have time?
Sure, Massimo.
So thank you, Sophia. Can you give us like an estimate for the first approach, the betweenness based on spanning trees, in terms of very large networks, say like 20,000 nodes? And I guess also edge density matters a bit for this kind of computations. How fast or how easy it is to run this?
Okay, I actually have some of those statistics here. Give me a second.
Wow, okay, so really, that's super accurate. Thank you so much.
So, yeah, well, by head, nowadays it's difficult to have all the information.
If she has the example with 20,000, I would
Wow.
think that you guys agreed with these questions beforehand.
Nothing was pre-staged. No.
Yeah, so so for 20,000 nodes, for example, yeah, it didn't scale so well. We have for example, and yeah, it's not so good. 2,000 nodes and 1,200 edges, 1,300 edges, it would take like around 1,500 seconds. Well,
okay, so a few hours.
A few
like
hours.
An hour.
But another thing that the student will do is actually implement this on Python and hopefully also in a more efficient way, because this is on Java, and Java, of course, although we were able to parallelize the weighted part, it's not the connected components part. It's still slower than if we do this with NumPy and so on. So I implemented the first version and it was much faster than the implementation that is now available. And hopefully we will also try to put this in NetworkX and iGraph and so on. So hopefully.
No, I'm asking because like I'm working with free association data, and in free association norms, the problem of filtering out spurious associations is very important. Since in these kinds of networks, distances are more important than redundant information, maybe we could use these techniques for performing a filtering that at least we know what's happening, what is it that we are doing. I really like a lot your interpretation of counting how many times that link is important for keeping the network connected in the minimum spanning tree. So that could be like a very powerful way to interpret better this kind of issues. And if you find a student, I have the data. So let's say that if you want to challenge, but these networks are a little bit larger than usual.
Yeah. No, I know that is for sure. I know that there are some implementations now that do some kind of an approximation calculation of the measure, but I only found for unweighted networks. So for weighted networks, it's really much more of a problem. And so to my knowledge, at least for the weighted version, ours is the only implementation. But yeah, for sure, that is also something that we would like to work and improve, because hopefully there will be more people using and more applications using it. Yes. Thank you.
Perfect. Thank you so much. Thank you.
Anyone last question?
I need to read the chat because I think there are interesting stuff there. it would be interesting to see the concept of allele applied on misinformation. Yeah, for sure. And spreading. Yes, and Chico also left the reference here. Thank you, Chico. I will, okay, it's open for me to then take a look later.
Okay, everyone. So if you don't have any more questions, let's close this session. Thank you so much again, Sophia, for accepting our invite, for your time.
For having me. Thank you.
And thank you everyone for show up and still up here.
Hope to see you soon in person.
Yeah. Bye-bye, guys.
Bye-bye. Thank you very much.
Thank you.